Ever puzzled over the complexities of genetic engineering? Today, we're unraveling one of its intriguing aspects: zinc finger nucleases. These powerful tools represent one of the first enzymes harnessed in the realm of targeted genome engineering. They're quite a marvel, you see. Born from the fusion of a zinc finger, a member of a naturally occurring family of transcription factors, to a restriction endonuclease, typically FokI 3. This union gives rise to a unique capability. The zinc finger domains recognize 3 base pair sequences on DNA. When linked, they can identify longer DNA regions, offering the specificity needed for precise genetic editing. And the FokI endonuclease. It works as a dimer, meaning double-strand DNA cleavage happens only at sites where 2 zinc finger nucleases bind to opposite DNA strands. Intriguing, isn't it? As we see, zinc finger nucleases play a pivotal role in the science of genetic engineering, but how do they function? Let's delve deeper. Understanding the functioning of zinc finger nucleases might seem daunting, but it's fascinating once we break it down. So let's get to it. Zinc finger nucleases, or ZFNs, are a marvel of nature and science. These enzymes possess a unique ability to recognize specific 3 base pair sequences on DNA. How they do this is pretty interesting. Imagine a chain of linked zinc finger domains, each one like a little biological detective scanning the DNA for its specific 3 base pair sequence. When these detectives find their match, they bind to it. This allows them to recognize longer DNA regions, providing an on-target specificity that's crucial for precise genome editing. But that's only part of the story. Enter FokI endonuclease. This unsung hero works as a dimer, meaning it requires a partner. The double-strand DNA cleavage happens only when 2 ZFNs, each armed with its FokI endonuclease, bind onto opposite DNA strands. This dual recognition system is elegant and efficient, requiring 2 ZFNs to recognize different, yet closely located, nucleotide sequences within the target site. This simultaneous recognition and binding not only ensure precise cleavage, but also naturally limits off-target effects, making ZFNs a reliable tool for genome editing. So, that's how these intriguing enzymes function. But what about their design and specificity? Let's explore. Designing zinc finger nucleases and ensuring their specificity can be challenging, but it's a task that scientists are tackling head-on. Diving deeper into the design process, we find that it's all about combining the right elements. 2 zinc finger nucleases, or ZFNs, are carefully engineered to recognize different yet closely situated nucleotide sequences within the target DNA site. This dual recognition and binding requirement is a clever safeguard that significantly limits off-target effects, making our genetic edits as precise as possible. Now, here's where things get a little tricky. In a typical ZFN, multiple zinc finger motifs are aligned in an array. Each of these motifs contributes to the overall specificity of the enzyme. However, they also influence each other, which can complicate the design process. It's a bit like trying to tune a piano, where changing one string can affect the others. This makes the design and selection of modified zinc finger arrays quite a challenge. The final hurdle in the design process is predicting the specificity of the end product. Given the complexity of the interactions between the motifs, this can often be akin to predicting the weather, difficult but not impossible. So as you can see, designing ZFNs is no small feat, but the rewards, precise, targeted genetic modifications, are well worth the effort. And there we have it, a brief yet comprehensive look at zinc finger nucleases, the unsung heroes of genetic engineering.